mice, transgenic and knockout mice, guinea pigs, rabbits, zebra finches, and non-human primates such as rhesus and bonnets. In 2018, he was awarded with Dr. K.R. Bharadwaj Award for Excellence in Laboratory Animals at a junior level by Professor S.P. Singh Baghel, Minister of Animal Husbandry and Fisheries, UP, at Animal Convention of Laboratory Animal Science Association of India at Lucknow, held in April 2018. He has been nominated as a member of Institutional Animal Ethics Committee of several institutes by CPC SCA, which enables him to ensure proper care and use and humane treatment of animals used in research, education, or testing in these institutes. He also ensures compliance to the guidelines, regulations, and policies framed by the same. He is also invited as a guest faculty in several training programs to give talks on various aspects of laboratory animal science and animal welfare, including CSIR, IGIB, JNU Delhi, NIAW Balabgarh, Banasthali Vidyapith Rajasthan, etc., and has also organized a number of such trainings at NDRC for postgraduate students and participants from all over the country. Sir, I welcome you to take the stage. If you were to walk when you talk, then you will just come on.
Thank you, Garima, for nice introduction. So I am thankful to Dr. Nitin and his team for inviting me to interact with the faculty members and other participants who have joined through online board. So this is a very useful workshop, or we can say training program, because use of animals in research, it's a privilege for us, because we have seen in COVID that there are so many drugs and so many vaccines which came into market that are initially tested in vitro, then on animals, and then after that human trials has begun. So animals always has given us good medicines, so we should use them with ethical responsibilities. Okay, so it's our responsibility that we should judiciously use animals in research. Okay, so for that purpose, this type of training programs and conferences are organized. So okay, so today we will discuss about the comparative anatomy of commonly used laboratory animals in research. So this is a very broad topic, because anatomy is very important subject when you are working with laboratory animals. So we will discuss in not in much detail, but we will cover most of the important things which you have to go through the for the animal experimentation. Okay, so we will not go in much detail for each and every species, because we cannot cover anatomy in 1 hour. So here are the contents what we are going to discuss in this lecture. So why do you want to learn anatomy? Then classification of animal groups, special features peculiar to each species, dentition of commonly used laboratory animals, and then organ systems like body cavities, respiratory tract, circulation, digestive tract, urinary tract, reproductive tract. Okay, so these are the terminologies which we use when we work with animals. Okay. So dorsal is the back or upper surface. Ventral is the belly or lower surface. Lateral towards the sides of the animal. Anterior, or we can say cranial, towards the head end. And posterior or caudal towards the towards the tail side. Okay. So medial is towards the midline of the animal. Proximal is closer to the midline of the body. Distal is farther from the midline of the body, and superficial is near the surface. Deep a distance below the surface. So you can see from the diagram below that all the terminologies has been explained. So then comes, why do we learn anatomy? Okay. So it is necessary to have a exact idea about the exact location of the organs. Okay, suppose for example, if you have to do cardiac puncture, if you have to withdraw from blood from the heart, then you must know the location of the heart. Okay, so you must know the location of organ, as well as you must know at what angle you have to insert the needle. Okay, so that you can directly go to the heart. And the second thing, if you have to do some surgical procedure, like you have to do adrenectomy, so you must know the location of the adrenal glands. You have to do pulmonary laparotomy, so you must know the location of the lungs. So these are the reasons due to which anatomy is important subject. Okay. And also the people who work in laboratory animal facilities, they must know the exact location of the organs as well as their normal structure, so that they can compare with the diseased conditions. Okay, sometimes there is some disease outbreak in animal facility, then in that case they have to do post-mortem examination. So in that case they must know about the location, their normal structure. Okay, so these are the important points which you have to learn before working with laboratory animals. Then coming on the classification system. So if you see at species level, so most of the animals which are used in laboratory animal facilities, they come under order Rodentia. So out of these animals, if we see at species level, only this mice and rats, they are mostly used in research. Okay, but other animals are also used, but mostly rat and mice are used. They cover around more than 75% of the research animals. So I have included birds as well as fish, because nowadays zebrafish is mostly used in laboratory animal facilities. Okay, so when we look at the rat, so laboratory rats, they are descendant of the Norwegian rat. So when we compare with the roof rat, which is a wild one, so you can see. So can we remove this upper thing or not? So when you see at this pictorial representation, you can see the tail of the roof rat, it's much longer than the hand plus body. Okay, but in case of laboratory rats, it is shorter. Okay. Also, the ears are large in case of roof rat, but in case of laboratory rats, they are not. Yeah. Okay, thank you. Okay, so the snout is also pointed in roof rat, but in case of laboratory rats, it's blunt. Eyes are large in case of roof rat, but in laboratory rats, they are small. So these are a few differences by which you can differentiate between roof rat and laboratory rat. Okay, so this is about distribution of animal species used for research, testing, and education. So you can see mostly mouse and rats, they are used in research, followed by birds, fish, guinea pig, rabbit, and other species. Other species includes the larger vertebrates like monkeys, dog, cats. So they are used very less. So what do you think? Why mouse is used mostly? Economic, yes.
Also, it's got a shorter lifespan.
So the most important is mice shares more than 95% of genes with human beings. So they are mostly used in research. And second point is because of their easy availability, their small body size, low cost compared to others, easy maintenance, high rate of reproduction. So their gestation period is short, 21 days. And the female gets pregnant on the day when she delivers pups. So within a short lifespan, you can get many animals. Okay, so and rearing cost, if you compare with other large animals, with the rabbits or guinea pigs, so rearing cost is very less. Okay, so you need to spend very less amount for housing and maintaining these mice. Then comes they can withstand inbreeding. So there are variety of inbred strains available in which you can do research. So what is the use of inbreeding? Why inbred strains are used in research?
The genes get particular.
Because there is a genetic similarity. Okay, so it minimizes the chances of variation in your experimental model. Okay, and this due to this you will get reproducible results in each animal. Okay, otherwise if the animals are outbred, then in each animal there are chances that you will not get the same results. Then comes there are animal models which are which is basically created for the disease which occurs in human beings like Alzheimer, Huntington's disease. So there are mice models available for these diseases. Okay, so we can easily create animal models of human disease, and by which means we will get accurate results and reproducibility also. Okay, then coming on dentition, so you can see most of these rodents, they have incisors and molars. They lack premolars and canines. And if you see on the fecal eating habit, so most of them are coprophagic. Okay, so they take this feces directly from anus in the morning time. Okay, because they are rich in nutrients, high in protein, vitamin B and K. So they directly take the feces from the anus. There is guinea pig also. So although the name has pig, but it is also a rodent. Okay, so and if you see on the this fish did not have teeth, they have this single pair of pharyngeal bones. So they did not have teeth, and they are not coprophagic. Okay. So then coming on teeth of rodents, so there are incisors and molars. So incisors are hypsodontic. They are open-rooted. They erupt and grow continuously throughout the life. So they are worn down only during mastication. So that is the reason we provide them with solid feed. Okay, we give them pelleted diet. Okay, so that when they chew the feed, when they consume the feed, there is a mastication of the teeth. Okay, otherwise there will be continuous growth of the teeth, and it will result in malocclusion. Okay, so after this, if you see that if you are providing the animal with the powdered diet or some soft diet, so there will occur malocclusion, and the animal will die after some time because it cannot consume feed after malocclusion. Okay, then comes molars. So molars are brachiodontic. They are permanently rooted. Say they do not continue to grow and used for grinding of the feed. Okay, so then coming on the external features of the rodents, so they have long tail, erected round ears, bright eyes, pointed snout with vibrissae, short legs, feet with 5 toes on each foot. The first digit on the front feet lacks a middle phalanx and is very small. The feet have walking pads and rudimentary sweat glands open to the surface of the foot pad. So basically, when we do this animal handling and restraining, then you can easily see the front paw has only 4 digits because this first one is a rudimentary, and the hind leg has 5 digits. This is an important structure in case of rodents because we have to withdraw blood. So this is retroorbital sinus. So you can see when you have to withdraw blood from retroorbital sinus, you have to insert capillary into the medial canthus. Okay, and with the help of capillary, you can collect blood in a tube or some other instrument. Okay, so this is just to show you the location of the retroorbital sinus for blood flows. This is about the gland. So rats has a horseshoe-shaped gland which secretes the red pigment called porphyrin. Okay, so if your animals are not in good conditions or if they are stressed, then there is a hypersecretion of this pigment. And this pigment, it stained around the eyeball of the animal. Okay, so you can easily see in the picture that there is a staining, red colored staining around the eye of the animal. So that means your animal is not healthy. It has some stress. Either you are not offering it good quality feed, your ACs are not working, or there are some noise in the animal facility, or there are some other stress factors. So in that case, there will be hypersecretion of this porphyrin pigment, and animal gets stained with this red pigment, and you can easily differentiate between a healthy and stressed animal. So this is the important gland. So usually there is a when the animal remains in normal condition, then also there is a secretion, but the animal wipes off with its forepaws. Okay, so that means it secretes usually this porphyrin pigment, but there is not much secretion. Okay, but in case of stress, there is a hypersecretion and it gets stained on the eyes of the animal. So this is just to show you the mandible salivary gland. So these are large glands in the neck of rat. You can easily, when you incise the ventral side of the neck, you can directly go to the mandibular salivary gland. So this is just to show you the mandible salivary gland. This is about the nervous system. So rodents, they have 2 cerebral hemispheres. So these are cerebral hemispheres, these 2 cerebral hemispheres. This is cerebellum. So you can see this is smooth. They did not have folds or grooves. But in case of human beings, there are folds and grooves. And they have 34 pairs of spinal nerves. So they have very simple nervous system. Then coming on the little bit about the hamsters. So these are used less than 1% in research, and most important feature is their cheek pouches. So these are used to transport food, nesting material, and pups, and also to store food. Then guinea pig. So these are mostly used in immunological research. Their stomach is entirely glandular, but in case of rat and mice, they have both glandular as well as non-glandular stomach. But guinea pig has glandular stomach. They have large cecum because they mostly consume this vegetables and other material. So for that purpose, they have large cecum for microbial digestion of the food. Left lung consists of 3 lobes, but in case of rat and mice, they have only 1 lobe. Male and female guinea pigs has 2 inguinal mammary glands. Coming on the Gerbils. So if you have to select Gerbil for any study, then you must know about the importance of Gerbils. So they are used for experimental atherosclerosis studies because they can develop high serum and liver cholesterol levels even with low cholesterol diets. They can produce very dry feces and small amount of highly concentrated urine. Rabbits. So rabbit has sebaceous glands under the skin of chin and around anus. They secrete pheromones to mark out its territory. So what's about rat and mice? They have sweat glands or not? They did not have sweat glands in the body, but they have sweat glands on the paws. Birds, they don't have sweat and sebaceous glands. Coming on the body cavity. So when we talk about body cavity, so diaphragm is very important. So diaphragm, in case of mammals, they have diaphragm, so it separates thoracic as well as abdominal cavity. But in case of fish and birds, they did not have diaphragm. So there is a no division. Okay, so when we discuss about the respiratory system, so mammals, they have lungs which are lobulated. Lungs have alveoli, so these are used for exchange of gases. But in case of birds, they have lungs, but they have air sacs also. Okay, so they do not have alveoli. They contains parabronchi. So air sacs, some birds have 7 air sacs, some birds have 9 air sacs. Okay, so these air moves in and out of the respiratory system due to pressure changes in the air sacs. Okay, so there are certain muscles in the chest which contract and they cause sternum to move outward. And due to this muscles, they create negative pressure inside the air sacs. So due to this negative pressure, air comes inside the respiratory system of the birds. And expiration is also a active process in birds. Okay, it is not passive, but in human beings, expiration is a passive process. Only inspiration is an active process in human beings. Okay, so there is contraction of certain muscles, and due to that contraction, they put pressure on the air sacs and air moves out of the respiratory system. So this is little about the respiratory system of the birds. So they have more efficient oxygenation than mammals. So fish, they have gills. Gills are specialized outgrowth from the pharynx. So gill lamellae increases surface area. Also, in fish, they have a one more structure called as swim bladder. So swim bladder is used for sound production, and it also helps in equilibrium with outer portion of the face. So birds, they have syrinx at the base of trachea, and mammals, they have vocal cords in the larynx. Okay, so this is about lungs. You can see the right lung is divided into 4 lobes, and left lung has only 1 lobe. But in guinea pig, there is 3 lobes. So you can see in this structure, this is heart, these are lungs, this is liver. Okay, a little bit more about the respiration in birds. So birds, they have 2 inspiration as well as 2 expiration. Okay, so this is cranial air sac. This is caudal air sac. So in first inspiration, air moves directly into the caudal air sacs, and in first expiration from caudal air sac, air moves into the parabronchiais. Okay, so there is exchange of gases. So then in second inspiration, the air from the parabronchi, it goes into the cranial air sacs, and in second expiration from cranial air sacs, it goes into the out of the body. So these are gills. So these are breathing apparatus of fish. They are highly vascularized, giving a bright red color. Operculum is a flexible bony plate, protects the gills. So water is inhaled through the mouth, passes over the gills and exhaled from beneath the operculum. Okay, then about the circulatory system. So fish, they have single circulation. So single circulation means blood, deoxygenated blood comes inside the heart and it goes into the lungs and get there oxygenated, and then it goes to the throughout the body. But in double circulation, heart in blood comes in heart, then goes to lungs, and from lungs it again comes to heart, and then it is supplied to the body. So fish, they have although they have 4 chambers, but only 2 are true chambers, atrium and ventricle. Other are they are false chambers. But in case of birds and mammals, they have 4-chambered heart. Okay, so this is just to show you the location of the heart. So the apex of the heart lies close to the diaphragm. So if you have to do cardiac puncture, so you have to initially feel the diaphragm in animal. Okay, so after feeling diaphragm, you have to insert needle at 45-degree angle, and when needle goes inside the heart, so blood comes in the syringe. Okay, so then you can withdraw blood. So it should be done under proper anesthesia. It should not be done under without anesthesia, because when you are doing cardiac puncture, it's a painful procedure and it should be terminal procedure. So many bird bones are pneumatic, means they have air sacs and they do not contain bone marrow. So RBCs are oval and nucleated, and thrombocytes are also nucleated. So this is about little bit more about fish circulation. That blood comes, deoxygenated blood comes in the sinus venosus. It is from the throughout the body, through the hepatic and cardinal veins. Then it goes into the atrium, and from atrium it goes into the muscular ventricle, and from muscular ventricle it goes into conus arteriosus that contains several valves and sends blood to the ventral aorta. So from ventral aorta, it delivers blood to the gills, where it is oxygenated and flows through the dorsal aorta into the rest of the body. Erythrocytes are oval and nucleated and use aerobic metabolism. This is just to show you about so this is sinus venosus. Blood is collected, then it goes into the atrium, then ventricle, and then to the gills, and it is oxygenated and spread to the throughout the body. So this is a single loop circulatory system. But in case of birds and mammals, there is a double loop circulatory system. Okay. So then coming on the digestive system. So fish has a very simple digestive system. They have bladder, liver, and pancreas. So there is little differentiation of the intestine. Birds, they have proventriculus and ventriculus. So proventriculus is through stomach because it secretes hydrochloric acid and other enzymes. And they have ventriculus. So ventriculus, also known as gizzard. So it is a muscular structure which helps in grinding of the food. So mammals, they have well-developed digestive system. And birds, they did not have teeth. So they also have one important structure called as croup. So whenever they take feed, that feed is stored inside the croup. Okay, so when croup is empty, it sends hunger signal to the brain, and then birds start eating again. this is about the stomach of the rats. So you can see there is a limiting ridge. So it separates the glandular stomach from the non-glandular stomach. Okay, so this limiting ridge, it prevents vomiting. So if you have to design some study in if you have to suppose some people say we have to do overnight fasting for to the animal before giving anesthesia. So there is no need to do overnight fasting in case of rodents, rat and mice, because they did not vomit. Okay, so this limiting ridge, it's not a actually there is no partition, so there is no physical partition, it's only a ridge. Okay, so it prevents the flow of the contents from one part to another part. They have large cecum for microbial fermentation of the food. Okay, rats, they did not have gallbladder. Okay, so if you are planning some study which involve gallbladder, so you have to select some other animal. So bile moves directly from the liver through a common duct to intestine. No, only rats, they did not have gallbladder. Mice has gallbladder. Okay, so this is about the caprophasic habit of the rodents. So food is taken from mouth, it goes to esophagus, then stomach, to small intestine, then cecum, then anus, and then return the feces to absorb more nutrients. So why there is a this coprophagic habit in rodents? Because mostly food is the nutrients are absorbed in small intestine, but the this cellulase enzyme is present in cecum. So when food passes through the small intestine, there is very limited absorption of the nutrients. In cecum, it breaks down more, and there is production of more nutrients. Okay, so it passes directly to the anus through the feces. So in the morning time, they take, because most of rodents, they are nocturnal, so they take their feed, more than 70% of their feed and water they take in night. Okay, so mostly they are active in night. So whenever they take feed, they will pass feces, and feces are then re-eaten again in the night on. Okay, not in the daytime. In the daytime, they pass solid feces. In the night time, they pass soft feces.
So is it like they don't have that absorbing villi on the intestine?
They have in the small intestine. So almost every animal have this villi in small intestine, but microbial fermentation, that occurs in large intestine. So initially food passes through small intestine, so there is less absorption of nutrients. Then it again breaks down in large intestine. Okay, so there is production of nutrients and vitamins in large intestine. But after large intestine, it does not go back to the small intestine. So it passes through the anus. So in that case, they re-eat that feces, and it again passes through the small intestine, and then there is absorption of the nutrients.
Do you have a sense to discriminate between first feces and second feces kind of?
Actually, this is what they have, means in night they eat most of their food. Okay, in daytime, when you go to the animal facility, you can see them sleeping and snoring. Okay, so you should not be confused that your animals have some problem, because in daytime they will sleep, in night they are active. So that is the reason when people plan some behavioral experiment, they have to reverse night and dark cycle, because we cannot go to the animal house in night. So what we do, because our working time is in the day from 9 to 5:30, so what we do, we reverse the cycle of the animals. Okay, because when they are most active, then only they can do good behavior. Okay, so we have to reverse, and in day their physiology, we reverse their cycle, so their night becomes our day and our day becomes their night. Okay.
So we have to make the rooms like.
No, you have to just change the timing of the light only.
But somehow there should not be any natural light.
No, there should not be any light. So there are timers available, so you just have to change the timing of the timer. Okay. So 12 hours light period, 12 hours dark period. Okay, so this is just to show you the digestive system of the bird. So this is beak, then food comes through the beak into the esophagus, then this crop, it's out pocketing of the esophagus, and it's located on the ventral side of the neck. So this is proventriculus, this is ventriculus. Okay, so this is then small intestine, then large intestine. So they have paired ceca. Okay, so then then this we have already discussed. Okay, so this is about the face. You can see the swim bladder, the stomach, the intestine. So there is no differentiation of small and large intestine. So they have simple digestive system. Okay. This is about the this is about the urogenital. They have kidneys, which are they have pair of kidneys, and it directly goes into the urinary bladder. So okay. So mammals, they have kidney, bladder, ureters, but in birds, their kidney is divided into lobules. So there is a lobulated kidney. Birds, they did not have urinary bladder. Ureter present that empties into cloaca. So fish, they have two long kidneys and bladder. So number of nephrons in the kidney, they are fixed at the time of birth. So we can see in humans there are 10 million nephrons, and in dogs 4 lakhs, and rats 1 lakh. Length of the tubules, they are species dependent. So we have seen in gerbils that they produce concentrated urine. That means the loop of fennel and the tubules, they are large enough that they produce concentrated urine and feces. This is about the kidney. So rodents, they have unipapillate kidney, so single papilla and single calyx. But in case of birds, they have kidneys divided into lobules. So you can see, so this is all kidney. So there are different lobules, and each lobule has cortex and medulla. Coming on the reproductive system, so mammals, they have well developed reproductive system. They have 2 ovaries, uterus, 2 testes in the scrotum, accessory reproductive glands. Birds, they have only left functional ovary, 2 testes in the coelomic cavity, no accessory reproductive glands. In fish, they have 2 ovaries and oviduct, 2 testes in the coelomic cavity, no or limited accessory reproductive structures. Mammals, they have prostate gland, seminal vesicle, bulbourethral gland. This coagulating and perpusticial gland, it is present in the rodents only, because when we start breeding of the rodents, and sometimes we have to do some timed pregnant studies. Okay, so you have to do some embryonic to study on embryonic stage 18, 19 or 20. Then in that case, when we start mating of the animal, on the next day we check for the plug. So this plug is due to this coagulating gland. So when they mate, that means there is a white plug is in the vagina of the female mice. Okay, so that plug indicates that mating has happened. Okay, so from that day, you can count, this is the day embryonic stage 0. Okay, and accordingly you can count that after 18 days you have to take the animal, and then you have to dissect out the pups.
this is about the urogenital system. You can see the this prepucial gland is present in the urogenital system of the rodents only, and there is no prepucial gland in the other animals. So this you can see the testes, reproductive system of the rodents. Testes, you can see left testis. This is the bladder. This is vasa deferentia. So mouse normally excretes large amount of protein in urine. Then about the uterus. So rats, they have bicornuate uterus, and each horn has cervix. So this is bicornuate, and you can see each horn has a cervix. Okay, so mouse has only a single cervix. And when you have to differentiate between male and female, so how will you differentiate? If you have to differentiate between male and female, in case of rodents, so you have to check the anogenital distance. So if the anogenital distance is more, then it is male. If anogenital distance is less, then that means it is female. So we will show you when we do practical, then we will check for male and female. So there is no reference. You have to compare. Means if you have, if you mix the male and female, then in that case you take 2-3 animals at one go, and you can check. You can differentiate between them. So that depends on your experience. So that we will show you in the practicals. Okay. So there are, because they have males, they have scrotum, there are testicles, but their inguinal canal is open. Okay. So when you pick the animal, you restrain the animal. Sometimes the testicles, they go inside the abdominal cavity. Okay. So without testicles, you cannot say it is male or female. Okay. So that is the reason that you have to rely on anogenital distance only. Large distance means male. Female. Yes. Okay, this is about the mammary glands. So rats, they have 6 pairs of nipples. Mice have 5 pairs. Hamsters have 12 to 14 pairs. This is about the birds. You can see the testicles, liver, spleen, epididymis. Okay, these are few reference books which you can consult for if you have to go in much detail about the anatomy of laboratory animals. So if you have any query, you can ask.
We are doing this dissection experiment we are doing this external organs we want to see. Then let's say if we are doing in mice and we are observing itself, and mice itself is very small.
Yes.
So how we will be able to use some aid or
no. There is no need to use some microscope or some magnetic. If you have to do some microsurgery, in that case, if you have to remove some internal structure, like if you have to do some ovaryhysterectomy, in that case you need some microscope. Otherwise, there is no need. For post-mortem examination, for collection of organs, you need not to have some device. With naked eyes, you can perform.
Maybe sir, I think I have not experienced. So one more thing, like if we are doing experiments, and generally for doing experiments we usually take only one sex, either sex. Okay, but by mistake, sometimes if we are doing a female, sometimes we take mice from the animal house
Hmm.
and by mistake they gave some pregnant mice.
Mhm.
So how we can differentiate that?
So, okay, so first thing is means when you indent animal, you have to specifically mention that you need either male or female. Okay, and also it's your it's responsibility of animal house also that they see the indent and they will give you the particular animal, either male or female. And also when you are taking the animal, you must check the animal that whether it is male or female. And second thing about pregnancy, if they have a gestation period of 21 days. So if the animal is in the E12 or E15 stage, then they start bulging of the stomach. Okay, so when you pick the animal from the tail, you can easily see the puffs in the abdomen. Okay, so you can easily differentiate between visually.
Or like some changes in weight or something.
Weight changes are there, but for that you must have initial weight. Okay, so you have to take a means whenever you are putting the animal for mating, you weigh the animal, and after 5 days or 10 days, when you are indenting the animal, then again you have to weigh the animal. So there is increase in weight when the animal is pregnant. Or you can do ultrasonography, but there should be availability of ultrasonography machine for that.
Something like HCG kind of thing we can do, like just for human beings.
No. That
Kind of thing.
So we have an online question from Mr. B. Verma. How to differentiate pancreas, especially if the animal is diabetic? The question is from B. Verma. He says, how do you differentiate pancreas, especially if the animal is diabetic? Must be the last one.
Okay, so basically, from which organ he wants to differentiate? Means pancreas are located in between the loop of the small intestine. So from which organ he wants to differentiate?
He is writing specifically for the pancreas differentiation, whether
Diabetic and non-diabetic.
Means is it possible
Between non-diabetic and diabetic pancreas.
So, as Dr. Vijay has said, so there is inflammation. So when there is inflammation, there is a change in color of the pancreas. So it.
The diabetic parameter to isolate it.
Okay. So for diabetic parameters, so it depends on research protocol, na? Again, whether it has some, so they can isolate the pancreas and they can see with the help of color changes, or they can do histology of the pancreas. So that is the only way. There is no other way.
So I have some questions. One question is there. So fish or the bird model can be used for what kind of a diseases?
So zebrafish is nowadays used for mostly neurodegenerative disease, because they have very simple nervous system. Okay. Also for some teratogenicity studies also. And other is birds. Bird model. They are good learners. Birds are good learners. Okay, so we have in our facility we have zebrafinch as well as crow. Okay, so zebrafinch have the ability that they sing song. Okay, so with singing, so people are doing research that how they learn singing from their parents. Okay, so basically that is a developmental studies. And with crows or with other birds which are clever, they can see the behavioral tasks performed by the birds, crows. Okay, so behavioral studies as well as developmental studies.
So is it true, like for using zebrafish we don't require
So it is nowadays it is required.
some presentation person was saying that using zebrafish is very easy.
So see, if it is, see, for example, if it is not required, but you need to put your protocol in the ethics committee. Okay, because whenever you are using animal, you must have ethical clearance. Even when you are taking samples from slaughterhouse also, then also you must have a ethical clearance, because journals, they ask for ethical clearance.
Audience, any other questions? Thank you, sir, for your detailed and informative presentation on comparative anatomy of laboratory animals, covering mice, rats, guinea pigs, hamsters, gerbils, rabbits, fish, birds, and other mammals. And the reason why the knowledge of this anatomy is important. For our next workshop round, I would now like to request Dr. Vijaypal sir to kindly start the protocol.
हाँ सर चल रहा है।
स्टॉप शेयरिंग मिनिमाइज कर लो। अपने लोग सुन रहे हैं वो?
हाँ सर सुन रहे हैं सर।
तो थोड़ा बंद कर, एक मिनट रुकिए सर।
आप म्यूट कर दो।
So Dr. Nitin will send you in a group to all online people a problem, and you have to fill the form B. You have to write a protocol for the problem that I have given you. Is it clear? I am giving you a problem. How many people? 82 people are there, participant online. So we will give you a problem that will be mailed to you, and that is popularly called as monkey studies. Somebody want to study monkey and some neural related thing. You have to fill the form B. Dr. Nitin will also circulate the form B. So we will let us discuss form B.
Should I put it on the screen, sir?
Yeah. Yes, this will go down like this, not moving. Okay, it's like this.
I'll do it, sir.
So this is the form B, where you have to write your protocol. Suppose I will go quickly one by one. Is it okay, Nitin? Okay, name of the establishment, where you are from, like FIEP, registration number that you have to ask from your institute, name and address of registration number of the breeder. In case if you are getting these animals from anywhere else, you have to write this. Why do you have to write this? Because you cannot buy the animal from the roadside. You have to buy animal from the registered breeder only. Remember that. You cannot buy the animal from the pet shop. Place where the experiment of presently kept, MIT, and date and duration of experiment. That you have to decide how long your experiment will go. 6 months, 1 year. If it will, you should also write a little bit longer, because sometimes animals don't come or some reagent is not coming. It's always advisable to write. Type of research that you have to write. So this was the question asked me by Nitin, what kind of research MIT can do. That also depends on what kind of registration your facility is. Whether your facility is a registration for collaborative research, basic research, or education research. In that one, suppose if education only, then you can do all kind of protocols that are related to PhD and M.Pharma. Now coming to your project and what is the title. Here the most important point is this, as I told you on the first day, that you have to only do the research which is useful and have some certain purpose. And this thesis title should be same if you are getting funding from DST. Issue is this, that you are getting funding from DST. DST is for somebody cardiology, and you started doing rheumatoid arthritis. And funding agency will never release the fund unless you have a ethical approval. And it is the responsibility of the institute that what you wrote to the funding agency is same as what you are doing. It should not be two. It's not that I have a project from DST, I will do. No. DST is only, as I have been asked by you, sir, first day. DST, funding agency, looking to the merit of the science. Ethics committee is looking to the all animal related issues. So you may have the funding, but you may not have the approval. So this should be matching. Principal investigation detail. Ma'am, can you go up a little bit? These are list of individuals authorized to conduct. List of individuals authorized to conduct a procedure under this rule. The benefit is this, that PI and the people who are doing the experiment should be trained. Here we are asking experience. How much experience in the lab animal experiment? Okay. So now onward you can say that we were trained for 5 days continuous faculty development program for lab animal science. So we can say we are experienced in that. So this is what we intend. We generally look to this on whether you are experienced or not. Funding source, as I told you, it should match what you wrote to the funding agency and what you are doing now. Duration is already done. You have to write exactly when you will start. Usually what we will write, I will start as soon as approval comes. No, not acceptable. You have to say which date you are starting. Describe the detailed study plan to justify the use of animal. Here we are asking what?
Sorry.
Yeah. Justify the use of. How you will justify it?
The outcome.
Yes. And this you already discussed. Anyone?
The outcome.
Outcome. You speak in mic.
The outcome of the project has to be something which has not been done before.
Yes. Statistically significant, very good. What else? Let's interact. I think it's good to interact rather than me speaking and you just. I think novel work. I think beneficial for society. But here they are asking why animals? Why not something else?
The similar study has not been
Done before. Very good. But where you will find?
on the database.
Database. Remember PubMed first day I told you? You have to write your key terms, diabetes, animal model, or if you have something like Chenopodium album, you want to see that from this plant I will have diabetes. Put these things in PubMed. Then what I would like to see here is generally, not in the science. We have gone to the PubMed database with these key terms, okay, and Boolean terms, and from these, total result was 10 or 20. And in this one, our study is not figuring, hence it is novel, and we are using animal. Why we are using animal? Because there is no established alternative available through this database and through these key terms. Please don't write it. We cannot have alternative. We cannot have the body need to be only animal and like that. No. Here you have to justify. You are going to the database. You search your term and you find that with these terms you have to use animal because alternative is not there. If you see alternative, then you can use alternative. This is very crucial. Don't cut copy paste. And for online participant, everybody have to fill. So we will see and then see. Next. Okay. Here you have to specify animal detail. What is the species and the strain? Species is mouse. Strain is like bulb, situation, blah blah blah. There are 2,000 strain. Here, when you say the strain and mice, you should know why it is. Remember, we discussed why this animal. Okay. Age and weight. This is very, very important. Why age and weight? Yes. Age and weight. And also reproductive system. You are doing an MIP and I am doing an IGIP. I should be able to reproduce your study by just seeing because you are specifying everything now. Age, weight, this and that. So there is no time about it. Gender. Somebody asked just only male. No.
Next.
You have to use both sex. So there are different guidelines, arrive guideline and all that one. And if you put only male or only female, which validity will be low?
Intrinsic.
Validity external. And your editor or your reviewer will also shout at you.
So in papers we are seeing like this, like whether sex, it's like only female they have used or male they have used.
Yes.
In similar studies.
But no longer now. If I will say you Arab guideline, they are always asking both the sex. Otherwise, what will happen? Usually, if you use one sex, you will get the signal which is high. But this will not go further. So, just to avoid the reproducibility issue, Arrive guidelines specifically say use both sex. As far as paper is concerned, I told you, paper may be for anything. But now ethics committee mostly say, if they are aware, that use both sex. That's why we are asking sex here. Number to be used. Number means? Yeah, sample size, not sex.
Sample size.
Yes. How many number, how many years, year-wise breakup. Number of days each will be housed is as you have mentioned, start and end date. Rationale for using. Why is animal usage necessary for this study? Okay, why it cannot be done into humans? Okay, nowadays, if you go to most of these universities, you will find most of them pharma theses are basically studying on Ayurvedic formulations. finding anti-diabetic or blah blah blah. Most of. Do you think it is okay to do such kind of a study?
Yes, sir, Ayurvedic things we are also using in day-to-day life.
Yeah.
So we don't.
Sir, they don't need traditional use, but we are generating data
For what?
for their traditional use.
Why? Who is asking you to create data?
Ultimately, we have to give benefit to human.
So, if you go to the Ayurvedic guideline, I can talk about that one, but they are bigger, no? So, Ayurvedic, which we have Ayurvedic, Siddha, Unani, and homeopathic and all. So, they also have a pharmacopoeia. Okay? They have also a pharmacopoeia. If they have some formulation which in their pharmacopoeia have indicated for that, you don't need to do any study. Because these are already proved. Why you are reinventing the wheels? Remember? So there are 28 books. Those who want, I just drop email, I can say. 28 books of Ayurveda are this Siddha, Unani and all. Where they have specified, okay, this is the formulation, this is the indication. You don't need to generate the data for that. If you are seeing some new indication, then only. But don't, these all what we are talking about is schedule wise drug or single molecule. Yes. So that is a different bundle of books. I don't want to open that. But remember, if there is a some this why animal usage necessary. If I am your ethics committee and you want a herbal formulation, I will ask where is the idea coming from? Have you looked into all this pharmacopoeia? If not, go and look. It is already done. Why you want to do it? Okay? Is this clear? Then comes the whether similar study has been conducted. Please don't write there as you feel. Go to database. We always try to be original scientists. That's not acceptable. Go to PubMed, go to Web of Science, go to Cochrane study, and then say these database have no such kind of information, hence it is not being done. Here comes the hit, sir. When you ask for say whether we can ask your science, we cannot ask your science. But if science is already been done, then we can say no. And we must have the proof. And also the in vitro model. In vitro model means something which can be done by 3D model and all. Why particular species selected? Species means mice, rat, rabbit, guinea pig, buffalo, turtle. You cannot justify कि हमारी MIT में mice available हैं, बिल्कुल इसलिए नहीं है. और बहुत easily है कि नर्दी देते चीज ब्रीडिंग करके बनाते. Easily available, easily reproducible, and also easy to handle. ठीक है? Not acceptable. Here you have to say, I am doing cancer model. In cancer model, I find genomic similarity like this. I am using this cell line, which is also there in the humans. Hence, this model is good for me. Scientific, physiological, scientific. Why estimated number? Not statistically significant. Not 6. This is emphasized. Here, many of our people who are sitting in ethics committee are also not aware of these issues. So if somebody says, no, I am the boss, you are saying 6, take 4. What you will do?
Show him the calculation. Why 6 is important.
6 is important. You calculate the name.
The calculation दिखाएंगे ना.
Yes, sir, we can ask why 4.
You cannot ask me, he is the boss. वो practical बात करो अभी थोड़ी हो गया. वो कहेगा अच्छा मैं मेरी बात से रिपोर्ट पूछ रहा है. No. अरे यार आपको बताया ना आप सो गए उसी में. मैंने कहा था कि
otherwise we will show the database difference.
No.
Or we will use that formula for the calculation of sample size.
In the rule of CPCA, it says 95% level of. ये तो बताया था कि भाई ये देखो आपकी किताब में लिखा है और आप लिख के दो मेरे को जो आप कह रहे हैं। हमारी किताब में क्या लिखा है, यू हैव टू हैव साइंटिफिकली वैलिड रिजल्ट, so it is minimum. So there is always a problem because we are not English person, we are अपना हिंदी वाला, so English person always maintain with the plain with the language. They say minimum number of animal, but statistically valid with 95% level of confidence. Okay, then you say the rule is that we have to use 95% level of confidence. Result should be valid. Valid means internal valid. Internal valid, do you understand? Yeah. इंटरनल वैलिडिटी इतना अच्छा बताया था यार, मेरी 2 दिन की छुट्टी खराब हो जाएगी, आप लोगों को भी ध्यान नहीं। हाँ, कोर्स है डिफेक्ट, पैरासिटामोल इज कोर्स डिफेक्ट, जो कोर्स है इनफैक्ट इस कर दिया कि इंटरनल वैलिडिटी ठीक है। What is internal cause? Cause means आपने कैंसर की दवाई दी, your दवाई is effect is simple part. Internal validity, external means can this go to human? So somebody who is a boss comes to you, you have to show the rule book. It's a minimum number likha, yes, but don't go to the literal meaning of minimum. Literal meaning you understand? Simple meaning. That's not the minimum number with a scientifically valid result and 95% level of confidence. The 3-4 बार दिखाओगे, रूल दिखाओगे तो शायद वो मान जाए। नहीं माने तो मेरे को मेल कर देना, ठीक है भाई। कहाँ तक थे? हाँ, तो similar experiment conducting partial establishment. You can say, oh, I am expert of cancer. We have done cancer model, but not with this molecule. Okay, so that is your duty. If yes, justify that this intervention which I am doing now is new, but rest of the things I am aware of. Have similar act being conducted by any other organization in same or in other living model? It just enforce the reference. If you think that this model is being established, like you want to establish the model in biology, which is been done in somewhere, you can say, okay, this is the model established there in animals, and hence is the reference. But I will do intervention. This is different. Describe the procedure in detail. Procedure in detail means experiment detail. Experiment detail मतलब कैसे लिखोगे आप?
कितने कंट्रोल हैं, कितने
हाँ, और ये नहीं लिखना ग्रेड केयर। ग्रेड केयर मतलब अब फिर तो आज भी चाय खिलाई मेरे को, ग्रेड केयर नहीं। ग्रेड केयर तो मेरा बहुत जबरदस्त रिक्वायरमेंट है, तो मैं कहूँ मिनिमम केयर या कहूँ 1 cup of tea after entering a mind. Then there is no confusion. So you have to write not great care. These things. Not nothing left to imagine. Great care, humanity. No. This anesthesia, this injection will lead to this.
So is disclosing our experimental protocol would be good at this stage? If somebody, for example, is going for intact patient or something, that be good at this stage?
Yeah, ma'am. What's your opinion? Vandita, yeah. Vandita, it's not a problem at all because we are not asking anything which is something to you. Suppose patent can be a compound. Procedure cannot be patentable. So in that you can say, I will not disclose what is the name of my compound. I will say XYZ, or you will say MIT 1, 2, 3 or blah blah whatever. But all these things are not patentable. You have to tell because these concern to the welfare and the experiment. What is can be your patent knowledge can only be, you can hide them, you can mask them by number.
And sir, in some most of the meetings, pre-meeting, people used to be signed that disclosure agreement.
So
9th point.
Describe procedure in detail. So in this one, If there is anything invasive, you are cutting or making some surgery or whatever, you have to decide. And in that one, you have to write which dose. Dose means how much you will give. Remember, there is always a limitation how much you can take and how much you can give. Not you can inject everything. So there is a limitation how much blood you can take and also how much you can inject. And also the site, which site? Site always important. If you are giving intravenous, the reaction is good. If you want the molecule to go very, very slow, go subcutaneous. If you want to very, very slow, then intradermal. Okay? Volume. If you are giving intradermal, you cannot give more than 550 ml. If intradermal, 500 ml. So these are the technical things that you have to only understand at that time. Blood withdrawal, you cannot take more than 10%. Okay? Mice में blood होता कितना? 1 ml. 1 ml का 10-20% होता 100 ml maximum. Where you will take blood from? Little बताया. Little under. आप फिर क्या है? मेरे जैसा बदमाश आदमी आ गया आपके पास कमेटी में, आप हो गए एनेस्थेसिया, लाओ एनेस्थेसिया दिखाओ। एनेस्थेसिया मिलेगा? ठीक है, हाइपरटेंशन में दोगे तो सब उठेगा, ये तो मिलर एनेस्थेसिया है। इतना शो दबाओ, क्या नाम सर आपका? क्या नाम है? यही तो बात है, डिस्कशन में मजा है। Vitamin, xylazine, and hypertension are general anesthesia, which are very, means these items don't come out of it. Whether they are very okay, do it, but we have to do something which is very good. So take isoflurane. We will try to show you isoflurane if possible. Isoflurane, no chloroform is not allowed. That is, we will teach. So isoflurane, put anyone unconscious, quickly collect. Basically, not to collect from here, but try to collect, because here if you are collecting with this capillary, you always have the chance that some of the glasses from the capillary are left inside. अगर देखो हमारे हाथ में थोड़ा सा जाता कितना दर्द होता है, और उस गिलास रहेगा तो एनिमल कितना दिखता है. So it is not generally advisable. There are multiple alternative methods out there. Tail vein, we can say theoretically, but never blood comes from the tail. Small can come, like if you want to do glucose, yes, you prick and the drop will come and you can do that. But it is practically difficult. So that's why you have to write all this.
Sir, I have read many articles in which they have given anesthesia right and even.
Yes.
So you are saying that it is not available.
Not very.
But then why they have?
पता नहीं क्या करेंगे, दुनिया चल रहा है, सब उल्टा सीधा क्या करेंगे? इतनी सारी चीज गलत नहीं है, चलती ना बात. हाँ, so science is evolving day by day. What is now best and good is now changed as per law. So ether and chloroform is not permissible, and for that we will discuss much in the classroom why they are not allowed or permissible. Okay. But good question, because most of the time people define, oh, it is already published. So published में तो सब कुछ है published. Ethics committee, that's why ethics committee is sitting. You have to adopt something which is latest and now, and can also justify. So this all you have to detail. Does the protocol limit the use of anesthetic or analgesic in the conduct of painful surgery? So everybody right and A not applicable. What we want here, that if you are doing anything painful, suppose if you are collecting blood from here, okay, we should use anesthetic, at least local anesthetic also, because isoflurane is general anesthesia, it will go. Local anesthesia means if you put this one like dental surgeon लगाते हैं आपको अंदर वो काफी देर तक वो गया रहता है.
Okay.
Yeah, that one same. Then anesthetic for eyes also comes. So try to use that kind of thing. If you are saying no, then we will ask why no.
But it will be already mentioned in the procedure before that, like we are giving the procedure in detail, sir.
Yeah.
Previously. So it will be mentioned there whether we are doing a surgical intervention or whatever we are doing, right?
Yeah.
So, so we can probably just say no and refer to the procedure above.
No, here also, in case if you are saying, I will not, then we will be concerned. If you are saying yes, I will use anesthesia, and then no problem. We'll ask you those. Many of the time, what is the issue basically? Scientist comes and tell you कि भैया मैं anesthesia use करूँगा ना तो मेरा सब disturb हो जाए, study खराब हो जाएगी, है ना? कहते ये सोच तो है कि नहीं है? अब उसके मैं आपसे क्वेश्चन पूछता हूँ कि आपने एनाल्जेसिया यूज किया, सपोज मेलोक्सिकाम, because you know the procedure is painful, you are cutting and doing some surgery. अगर हमारे भी सर्जरी वगैरह कुछ होती है तो we are using anesthetic, antibiotic and analgesic for a week. Because it's very painful, you have cut the tissue and disturb the tissue. So, if you are using this meloxicam, you know its mechanism? Yes or no? COX inhibitor, you know it. So, you know also the consequence, what could be the consequence. If you don't use it, and you don't know which are the pathway and parameter and other things got disturbed, because if it is pain, yes, pain receptor, stress hormone, corticosteroid, and everything will be upregulated. So how many things will be upregulated? You are not aware.
Noise level increase.
Yes. So just to say, okay, no, I will not use it because it is disturbed. Rather use something which you are aware. Okay, this is the thing. This is COX inhibitor. Will only have the half-life of this. This is so no issue. Are you agreeing with me or not? Rather than not using, and if you say, okay, I don't want to use it, like you want to immediately do something, then decapitation out, you have to have a strong scientific justification. Why are not you using it? Will survival surgery be done? Remember I told you, is multiple surgery allowed on a single animal? No. So here we are careful that you say, okay, if animal is okay, then I will do again another surgery on them. No, it's not usually. If yes, the following. If you have surgical procedure, you have to describe, name the qualification and experience level of personnel in bold. If you are doing surgery, we want you to have a veterinary doctor or somebody who is trained in laboratory animal science who can do surgery. Okay, because this is not, we consider this is easy, but it's not so easy. Post-operative care, remember, if you are doing middle cerebral artery, the first 24 hours you are giving how much time? 95%. You have to sleep there only, animal house mein. Okay, for next 72 hours, very less time. Just go and see. Once they have recovered, you can go and come back and see. That's the so if surveillance surgery to perform more than once on a single animal, so usually we will say no, it is not allowed. Describe post-experimental procedure. Scope for reuse. Generally, this is for larger animal, not for smaller animal. Rehabilitation is also for larger animal. Mouse, rat, rabbit, guinea pig, no rehabilitation is needed. For other animal, just you can describe method of euthanasia. Euthanasia is okay. 5 animal, same cage, go and take to the CO2 chamber and euthanize. It's not that. So, Dr. Nitin, we will show also one movie on last day. So it's not that animal भी वही है, euthanasia वही चल रहा है. एक student blood collect कर रहा है, एक student tissue collect कर रहा है. Not? Is it okay? Because I will show the movie how they talk and how the experiments will be disturbed. So euthanasia will be single, where no other animals are there, and it should be as quick and as painless as possible.
So in case if you are not having the COT check,
Serpentine sodium. Very good. Cervical dislocation if you are trained. Many of the time you have done cervical dislocation and will still alive and pain, feeling pain, and you are cutting on the animal. Okay. तो ट्रांसपोर्टेशन मेथड की अगर आपने मेरठ मेडिकल कॉलेज से तुम्हें लेकर आए कितने दिन रेस्ट कराएंगे, क्यों कराएंगे?
क्वारंटाइन कराएंगे
1 week क्वारंटाइन, क्यों? आप तो कह रहे 1 week, आपका बॉस कह रहा है कि जल्दी करो, टाइम खत्म हो रहा है, अगले दिन लगाओ एक्सपेरिमेंट, क्या बकवास कर रहे हो? फिर लगा दोगे? नहीं, पर वो कहने में लगाना ही पड़ेगा, वो तो ऐसा नहीं है, पर फिर भी कुछ ऐसी बात कह दो कुछ साथ मान जाए. Yes. If you see that animal, if I have come from Najafgarh to here, because I always travel, no problem, I know. Okay, horses are always very good traveler. People are taking abroad, they are having a horse carriage and they always travel, so there is no problem. Mouse don't travel, so it's a big stress to them. If you see the mouse which is traveled, at least for a week they are hypertensive. No need. Yeah. आप कंफ्यूज क्यों करें? ट्रांसपोर्टेशन, हाँ, आपने कहीं दूसरी चीजों से लाए हैं तो भी है ये. अगर आपका सिमिलर सेम जगह सेम खाना है सेम पीना है, ठीक है ना, नो प्रॉब्लम. बट हेल्थ मॉनिटरिंग एंड ऑल दिस वन आर डिफरेंट इशू. ओके ना, because initially when you are new to the system, so first day I came, you were different. Second day I came different. Third day will be more friendly. And if I come regularly, then also very. Then this comes usually hazardous. If something like this you are doing, then you have to take institutional biosafety clearance also and submit the same. This is not much better to take a biosafety clearance. Now coming to you, this one, you are a PI. Okay? You are signing a declaration, and that declaration you will read one by one.
Okay, sorry.
I certified that the research protocol submitted is not unnecessarily duplicate. Unnecessary, remember? पहले दिन वाला necessary याद है नहीं है? हैं जी? एक्ट में लिखा होता है. हाँ, तो लिखा हुआ लेकिन उसका कैसे justify करोगे? Is there a suffering? Is there a necessity or mens era not required as you more started? Okay. I certified that I am qualified and have experience in experimentation on animal. This is thanks to the Nitin that all of you will see now with the raising your collar that yes, I am okay and certified and trained. For procedure listed under this that I have reviewed the pertinent scientific literature and found no valid alternative. Alternative, remember I told you how to search. Go to PubMed, put key terms, MSS subheading, and there you will find only those alternatives which are done. But remember, if it is regulatory, they may not be acceptable. Regulatory is totally different. When it is a regulatory research, then most of these things, sample size does not apply, because the regulatory says this weight, this sex, this number, everything they say, and these guidelines are called as OECD test guideline. OECD test guideline. GLP you might have studied, no? Organization of Economic Cooperation and Development. There you cannot say, oh, 6Y, because that is regulatory. I will obtain approval from IEC in Form B that we have discussed before initiating any change in the study. No change can be done by you. Initially you have written biopsy, then you thought you don't have biopsy. Let's take rat. That is a change, and change in approval again. Even a minor change needed approval. If certified that I will not initiate a study before approval. The purpose will be initiated upon review and approval of scientific by appropriate expert body, institutional scientific board committee, funding agency. Yes, that is always there. Either somebody is funding, but that should not change. Funding agency would submitted something else, and here you are doing something else. That is the institutional responsibility. I think Nitin, usually every institution have a kind of a pre-committee where like sir have a patent in cancer. Have he written the same, means funding agency written the same thing, or he changing? That is the responsibility of the institutional. So you can have a institutional scientific advisory, internal committee, screening committee, pre-screening committee, something like this, whatever. Okay. I certify that I will submit appropriate certification of review and concurrence of study mentioned in point 14. Okay, that we have discussed. I shall maintain all record. What record? Form D. Form D means how many animal approved, how many used, and all. This is your responsibility. Usually you say, meeting animal house से रिचार्ज हो जाने में या स्टूडेंट जाने, ठीक है? ये आपकी जिम्मेदारी है, आपका साइन है नीचे. Okay. I say, but I will not initiate before approval. This is very clear. I certified that ensure the rehabilitation policy are adopted. This is only applicable when you are using larger animal, not for small animal, and you are putting on this signature and review. So, dear friend, those who are online and offline, this is the form that you have to fill and submit. So, this form will be shared with Nitin. How he will share? I will share the from online. So I will share with you two forms, form B, which you have to fill, and a problem which I give to you. Remember, a problem I will give to you, you have to read, and based on that, you have to write a protocol and submit to the Nitin due course. I think probably by tomorrow evening. So with these words, I say thank you very much.
Here's a question.
Okay.
Who will be the PI? Suppose I am doing any research or any animal experiment with my student. So who will be the PI? Myself or my student?
You. PI is a responsible person. And if anything goes wrong, the consequence will be with even the institutional head. He is the ultimate head. So he should be always kept in loop. Like your institute is head is principal or director, whatsoever. If something goes wrong with the animal experiment, he is responsible.
So is it on the qualification of the person? Is it on the
Qualification of the person?
What?
Being the PI, yeah. Experience and qualification? Not necessarily.
No. Usually those who are doing scientific research, they are qualified, but there is no qualified mark which have been said, because like Sita people might be BMS, sorry, BIMS or something. So nothing like that. Qualification is not the mark, but they should be trained in experiment as we have discussed in Form B.
Since the students are also getting training during this courses, so can we write their names to the?
Yes. So there are two. Can you go, ma'am? So first one. Yes, here is the principal investigator. Here, all individual. So here the students will come. All student. And if you think some student probably will go, so try to put some extra student. But when you are putting their name, ask them whether you are trained. If not, get some training.
If two investigators are there, the principal of the two investigators.
Yeah, it can be. Yeah, they can be there. That's not a problem. It is not basically hard. It's not basically to create hurdle. If you are 2, 3, 4, no problem. But whosoever you may be, there should be one PI which is responsible. You can write co and student, but one, you cannot say, no, he also co, he's also co, then nobody is responsible. Anything else? Anything from online?
No, I don't think so.
Can you put this online? जशी conclude.
Thank you, Vijay sir, for explaining the form properly to us. So we would be giving a problem very soon to all the online participants. We request you to fill the form, which is already shared on the Telegram group by Mr. Salman. So we will be discussing this form tomorrow morning, and we'll see if you are on the right track or not with that. So are you taking also? Okay. So we'll break for lunch and get back